everyone. In this video, we're going to describe the basic functions of all of the organelles and the nucleus. So the nucleus actually really deserves its own video and it deserves a lot of time, but most of the functions that we're going to discuss with regard to the nucleus are not going to be a major focus of our class, and so therefore we're going to do them later on. And so this is really just meant to be an overview of all the organelles so that we can sort of understand why they matter in later cell types. So we remember that the cell membrane is a phospholipid bilayer, but for the purpose of this video and many of the cell videos that we're going to draw, I don't want to draw that entire membrane in a large ring as many times as we're going to need to draw it. So we're going to just draw a line, but I just keep in mind for this video that line represents that phospholipid bilayer. And we're just going to fill in the nucleus and some of the organelles. So remember that this is also the model cell. So this particular cell is going to have everything. But when we talk about like special cells like neurons or special cells like contractile cells that are found in the heart, we're going to specify which organelles are plentiful and which are strikingly absent. So the nucleus is the control center for cell operations. So think of it as sort of like the brain of the cell. It stores information that's needed to synthesize proteins, and as I mentioned, that's a process that we're going to explain in a separate video. Around the nucleus is a structure called endoplasmic reticulum. So basically the endoplasmic reticulum is abbreviated ER, and it's a network of membranes that are attached to the nucleus. And a lot of that is because they have functions that involve communication with the nucleus in some ways. So there are two types of endoplasmic reticulum. There's the smooth endoplasmic reticulum, which is abbreviated SER. The smooth endoplasmic reticulum is responsible for synthesis of carbohydrates and lipids. It's responsible for storage of glycogen in liver and skeletal muscle. As an aside, glycogen is stored also in granules in the cytoplasm and a couple of other places. So it's not the only way that we store glycogen, but it's one of the ways. And then also the smooth endoplasmic reticulum is responsible for detoxification and inactivation of drugs. One thing that we can start to appreciate really early on is that many of the things that we know the body does will start to attribute to what cells do and what the structures inside of cells are doing in order to make those processes happen. The other type of endoplasmic reticulum is the rough endoplasmic reticulum, or RER, and it's considered rough because it's covered with ribosomes. So ribosomes are another organelle that we'll describe in a moment, but the presence of ribosomes on the endoplasmic reticulum gives them a rough appearance. The rough endoplasmic reticulum is responsible for the synthesis of proteins, and these proteins can be shipped to the Golgi Apparatus, which is another organelle we'll describe in a moment. So before we get too ahead of ourselves, let's talk about ribosomes. So ribosomes are also found in the cytosol. So we consider ribosomes as fixed, meaning they're attached to the RER, or they're free, meaning they're found in the cytosol. Ribosomes are also responsible for protein synthesis, and they actually they utilize a special type of RNA to actually make the proteins into the linear chains that we mentioned. Next, we have the Golgi apparatus. So we also mentioned that the rough endoplasmic reticulum is going to make proteins and then ship them to Golgi. And the Golgi apparatus is responsible for receiving, packaging, and shipping of the proteins that come from the RER. So basically, we could think of the Golgi apparatus as like a post office. And so something is going to arrive at the post office, and in the post office, it's going to get a mailing label, and that mailing label is going to help that particular protein know exactly where it's going. So it's giving it directions, it's giving it a destination. We'll just do a quick little animation here. So we see the protein, it's coming in a transport vesicle. So part of the membrane from the rough endoplasmic reticulum is going to enclose this protein, and then it will break off and travel to the Golgi apparatus. And the Golgi apparatus is also a network of membranes, so a similar thing is going to happen. And then when it's done, it will leave in the form of a vesicle again, and now it's a protein with a destination. So I know that the concept of what just happened here is kind of abstract, we're going to actually talk about vesicular transport as one of the examples of membrane transport, and we'll be able to animate this process a little bit more clearly. So don't feel obligated at this moment to fully appreciate what's happening when we transfer things from one organelle to another in a vesicle, because we're gonna spend a video talking about that. So for now, just sort of focus on understanding the basic function of each organelle, so that later on we can develop a full understanding of what's happening inside the cell at any given time. Okay, another example of something that can be released from the Golgi apparatus is a lysosome. So a lysosome is a vesicle that contains digestive enzymes. And so you could think of, maybe the rough endoplasmic reticulum is synthesizing the digestive enzymes, and then they're inside of this vesicle. And also inside this vesicle, along with those enzymes, is going to be a harsh environment. So probably something with a really low pH, something that's kind of acidic, something that is going to cause the breakdown of organic molecules. So organic molecules that are broken down in lysosomes can be recycled. The parts can be utilized to make new organic molecules later on. So this is necessary for sort of making sure that all the parts of the cell are working in good working condition. And if something is breaking and needs to be recycled, it can be broken down, and then the cell will utilize other structures to synthesize the replacement. So maybe a protein that's found in the membrane, maybe a piece breaks off of it or something, or it's starting to unfold, it's starting to get tired. So now it will be digested by the lysosome and replaced by a new protein that can be inserted into the membrane. One thing that might be interesting is that when muscle cells are inactive for a prolonged period of time, lysosomes can gradually break down the contractile proteins found in the muscle. So it sort of will explain atrophy. So like why a muscle that is big at one point in time, after much inactivity, why it starts to decrease, because parts of the cell are actually being deconstructed by the lysosomes. Another example of something that could come from the Golgi apparatus is a secretory vesicle. So something that's going to be, it could contain some type of material, probably a protein that's going to be moved into the extracellular fluid. Again, this is really an example of vesicular transport, so we'll see this in greater detail coming up. Another structure that's similar to the lysosome is the peroxisome. It's specifically responsible for breakdown of fatty acids, and the byproduct is hydrogen peroxide. So hydrogen peroxide is actually toxic to cells, which is why it's contained in the lysosome. Similar to that, maybe you remember, having scrapes and stuff as a child, and then having hydrogen peroxide be put on the skin, and it bubbles and it burns and stuff, and it actually can destroy healthy tissue, in addition to any bacteria or toxins that might be trying to get into the body as well. In order to ensure the hydrogen peroxide doesn't become toxic to the cell, there are other enzymes that are contained within the peroxisome that will degrade hydrogen peroxide into water and oxygen. Mitochondria are actually really interesting because they are very complex. They have their own DNA. We call it mitochondrial DNA. And it actually is inherited only from mothers. So any DNA that you have in your mitochondria is from the female contribution to the baby. And the mitochondria are responsible for cell respiration, which basically is the process of making ATP. When I think of mitochondria, I think of the 6th grade definition, which is that it's the powerhouse of the cell. So any cell that needs to do any activity needs to have mitochondria, because it needs to make the ATP in order for any cell functions to continue to occur. So anything that requires energy needs to have mitochondria. Not all cells have mitochondria, but the ones that don't on purpose, because it would inhibit their ability to perform their vital function. So an easy example of a cell that does not have mitochondria is a red blood cell. A proteasome is similar to a peroxisome. It contains enzymes called proteases, and it's responsible for digesting damaged or denatured proteins. So remember that denatured means that the protein has unfolded and it's probably not able to fold back up again. So we need to recycle those proteins as well. I'd like to make a couple modifications to the membrane. So one structure that is kind of like a membrane modification is microvilli. So instead of having a linear membrane, we can have a membrane that contains these mini folds, or kind of like finger-like projections. That's how they're often described. And basically what we're doing is we're increasing the surface area of the membrane. So commonly microvilli will be found like in the digestive tract because we want to absorb all the nutrients through the cells in the digestive tract, and we want to make sure that everything has good access to the membrane. So if we can scrunch up the membrane a little bit and have sort of more membrane per unit area, then we can increase the rate at which things can cross through the membrane into the cell. Another membrane modification is cilia. So cilia are actually microtubules, which we're going to describe in a moment. But basically, cilia are like they're like hair-like extensions of the membrane, but it's actually a different organelle that sticks out of the membrane. The cilia beat rhythmically to move fluid across the cell surface. So a good example for cilia would be in the respiratory tract. Actually, cilia are found in the respiratory tract, the urinary tract, and the reproductive tract. So not everywhere, not all cells, but definitely those cells that are exposed to the exterior. But let's just talk for a second about respiratory. So what happens when you have, so in the respiratory tract, there is a constant secretion of mucus, and the mucus is responsible for trapping particles, things like dust, dirt, things that we don't want to get into the respiratory tract. In addition to that, any bacteria or toxins or things that really we don't want to breathe in, they'll get trapped in the mucus. And then the cilia is going to beat the mucus, sort of like help it flow across the lining of the respiratory tract. So the idea is to get it to a point where it can be, the toxins or the dirt or the particles can be coughed out, or they can be swallowed. So the direction of movement is towards the mouth and the nose, so towards the exterior, and we want to just sort of make sure that stuff doesn't get trapped in there. So what happens with smokers is smokers actually have chronic cough. Their cilia die when you're a smoker for a long time. The cilia die, and then the mucus is not as well swept up the throat, and so there's more coughing because the mucus is less able to be moved out of the respiratory tract. One positive note is that oftentimes for those who quit smoking, the cilia can actually grow back. Next, let's mention centrioles. So centrioles also have a role in cell division. So we'll do a separate video on cell division, and we'll talk about centrioles. We'll talk about how cell division actually happens, and we'll spend a little time talking more about the nucleus in that section as well. Not all cells have centrioles. The cells that don't have centrioles, though, are not able to reproduce. Examples of cells that do not have centrioles include mature red blood cells, skeletal muscle cells, cardiac muscle cells, and neurons. So no ability to divide, no ability to reproduce. Next, let's talk a little bit about the cytoskeleton. So the cytoskeleton has four major components. First is microfilaments. Microfilaments are actin, and actin is a protein. It's also called thin filament. The actin are responsible for anchoring the cytoskeleton to the cell membrane. So they have a major role in the scaffolding of the cell. And then in addition to that, we will also see in muscle cells, they have a major role in muscle contraction. Next, we have thick filaments, also called myosin. So myosin is another protein, and in muscle contraction, actin and myosin interact. And so we'll spend a lot of time talking specifically about actin and myosin. There are also intermediate filaments that are responsible for strengthening the cell. And microtubules, which are responsible for many things, including cell division, including they make up cilia, including they're responsible for supporting the cell, and they're responsible for movement of certain materials within the cell from one part of the cell to another cell. Okay, and then very last, we're going to just try and squeeze it in here because we're out of room, is the flagella. So the flagella is a tail. It has a whipping action, and it actually is responsible for cell movement, propelling the cell forward. The only cell type in the body that has flagella is the sperm cell. Okay, so that's a lot. So we're gonna wrap it up there. I think that if there's one really important take-home message, it would be that not all cells have all these organelles. But when we mention the organelles to describe a specific cell type, it really means that that particular organelle or those particular organelles are critical to the functioning of that cell. So we're going to see a wide variety of cell types, and none of them are going to look just like this. So anyway, that's all for now. Hope you enjoyed it. Thanks for watching.